Today we are going to discuss about biogenic amines and chemical assessment of quality in fish. Biogenic amines are biogenic substances with one or more amine group. They are basic nitrogenous compounds formed mainly by decarboxylation of amino acid and also by transamination of aldehydes and ketones. Biogenic amines are organic bases with low molecular weight and are synthesized by microbial, vegetable, and animal metabolisms. The first one is histamine. Histamine is one of the most major biogenic amine found in fish, and histamine is mainly found in fishes comes under the family Scopidae. Example for that fishes is mackerel, seafish, tuna. The body of these fishes contain a large amount of free amino acid histidine. By bacterial decarboxylation, this histidine is converted into histamine, and once the histamine is formed, it is a major spoilage indices found in fishes. Endogenous bacteria, that is Proteus morgani and Enterobacter aerogens, are mainly responsible for the conversion of free histidine into histamine. Endogenous histamine is responsible for physiological functions related to local immune response, gastric acid secretion, and neuromodulation. The second biogenic amines are cadaverine and putrescine. These are the two other most commonly found biogenic amines in fishes, and they are also produced from amino acid. The precursors of these two biogenic amines are lysine and ornithine respectively. In case of fresh fish, the amount of these biogenic amines are very less. In case of spoiled fish, the chances is very high. Next is tyramine. Tyramine is another biogenic amines found in fish, and it is produced mainly from the amino acid tyrosine. The fresh fish contain very or little tyramine content, but in case of spoiled fish, the amount of tyramine is very high. Next is chemical assessment of quality. In chemical assessment of quality, the various spoilage products are quantitatively estimated and evaluated with sensory characteristics. These are compounds in fish muscle produced by the autolytic enzymes, putrefact microorganisms, or by chemical reactions like lipid oxidation. The examples for the various spoilage indices mainly composed of total volatile bases, nucleotides, and lipid oxidation products. In case of total volatile bases, these are mainly nitrogenous compounds comprised of ammonia, TMAO, TMA, DMA, etc. In case of nucleotides, these are produced mainly by ATP degradation. ATP degraded into ADP, then AMP, IMP, and hypoxanthine. In case of lipid oxidation products, they include peroxide, hydroperoxide, and aldehydes, etc. First one, total volatile bases. As earlier said, these are basic nitrogenous compounds and mainly comprised of TMAO, TMA, and ammonia. It is one of the major spoilage indices used in quality assessment of fish. If the total volatile basis nitrogen is in the range of 35 to 40 milligram, it is acceptable. Beyond that limit, we can consider that the fish is spoiled and not used for human consumption. Second one is trimethylamine. Trimethylamine is also total volatile basis and a major nitrogenous compound found in fish. If the level of trimethylamine or TMA in between 10 to 50 milligram, we can accept the fish. But if the limit is more than 10 to 50 milligram, we can regard as the fish is spoiled and not used for human consumption. Next is histamine. As earlier said, histamine is produced from the free amino acid histidine by decarboxylation process. Proteus morgani and Enterobacter aerogen is responsible bacteria which convert free histidine into histamine. Once the histamine is formed in fish, it becomes a toxic substance and causes spoilage known as comroid poisoning. If the histamine range is between 15 milligram or per 100 gram of fish, we can accept a fish, but beyond that, we can reject it as the spoiled fish. Next is indole. Indole is a spoilage index mainly used in case of shrimps. If the range is in between 25 gram microgram per 100 gram of prawn meat, we can accept the prawn. Beyond that, we can reject it as a spoiled shrimp. Hypoxanthine. Hypoxanthine is also a spoilage indices. It is a ATP degradation product. ATP degraded into first ADP, then AMP, then inosine monophosphate, then hypoxanthine. Hypoxanthine is a major spoilage indices, and we are using as a chemical indices to assess the spoilage of fish. If the range of hypoxanthine is more than 2.5 micromoles per gram, is rejected as a spoiled fish. K value. Next is spoilage indices is K value. K value can be defined as the ratio sum of inosine and hypoxanthine to the total concentration of other nucleotide present in the fish. If the K value of a fresh fish is between 20 to 25, and in case of spoiled fish, the K value ranges in between 50 to 60%. Thank you.